que son las de Ray Kim, pero también Mauricio todavía tiene una plática más con nosotros acerca del tratamiento de las recurrencias y la importancia de las resistencias en la era de los medicamentos pangenotípicos. Mauricio, please.
Thank you, Arturo. Es una situación para mí sumamente extraña estar en mi país hablando en inglés. Me pidieron que lo hiciera y pues lo haré. I'll share with you a talk related to treatment of hepatitis C recurrence, the importance of drug resistance in the DAA era. Now, if you see the title of this lecture, you can see three different themes. One of them is Hep C recurrence. The second one is drug resistance. And the third one is the DAA era. So let me talk first about what is the DAA era. Then let me touch about what is drug resistance. And let me finish with what is the treatment of hepatitis C recurrence. And I will try to put together these three areas to cover the topic of the title that was assigned to me. This is a cartoon that shows you the different eras of treatment of hepatitis C. As you can see, in the 1990s and all the way to 2011, we started to use interferon. That was followed by ribavirin and then pegylated interferon. And that era was an era that was not very effective in terms of the development of a sustained virologic response. Only about 40 to 50% of the patients that were treated responded to treatment, and the treatment was very effective only for patients with genotype 1A. In 2011, we saw the emergence of the first group of antivirals that have a direct action against the virus. And the first group was represented by two NS3/4A protease inhibitors, telaprevir and boceprevir. And that was followed by the beginning of studies that use other drugs, including cimeprevir and some of the initial investigation of sofosbuvir. In this early era, or the early DAA era, the overall sustained virologic response increased to about 65 to 75%, but again, we were treating mostly patients with genotype 1A. So it was very clear, based on the results that we obtained in the pre-DAA era, that we needed much better medications, and it became very clear that we needed much better treatments, more efficacy in the early DAA era, and more importantly, to probably expand the treatment of this disease to other genotypes. Since then, we have seen a tremendous revolution. This is an incomplete slide that shows you the progression of investigation in the DAA era since 2014, when sofosbuvir and ledipasvir appear in the market and approved by the FDA. Now, not only the amount of research and the new drugs that are available has been expanded tremendously, that the pace of this revolution has been such that today some of these medications have already disappeared or are disappearing. And that is only telling you how fast we're moving in this field. While the use of pegylated interferon and ribavirin lasted for about 2 decades, the current medications against hepatitis C have been surviving just a few years. This is a cartoon of the genome of the hepatitis C virus. As you can see, it has a non-structural and a structural region, and the non-structural region contains all the different enzymes that make the virus to be pathogenic. When we are using medications of the DAA that are working in the NS3/4A non-structural region, we call them protease inhibitors, and those protease inhibitors have a suffix previr just like telaprevir or boceprevir or paritaprevir or grazoprevir. When we are utilizing medications that work on the NS5A area, we call them replicative complex inhibitors, and in those patients we use the suffix -vir, like ledipasvir, velpatasvir, elbasvir, rusasvir, etc. And when we are attacking the enzymes that work on the DNA, RNA-dependent polymerase of the virus, or the so-called polymerase inhibitors, we use the suffix buvir, like precisely sofosbuvir or the sabuvir or others. And this is a very simple way for someone that is teaching these medications to learn and to remember them. So this is the background of the DAAs. So let's talk about drug resistance. Drug resistance has a very specific nomenclature or a very specific terminologies. And I decided to bring some concepts for you to learn them or perhaps to just remember them. One of them is called variant. A variant is a variant or a virus that grows better than wild-type viruses. Those are called the resistant variants, and they grow much better in the presence of DAAs. So we don't call them resistant-associated variants or RAVs. We call them variants or resistant variants, and we don't use them normally because in order to detect them, we need to do studies in vitro. The next term is substitution. When a virus that is sequenced from a patient has specific substitutions, that is, the RNA chain changes in sequence, you develop a resistant associated substitution, and we call them to those RASes. Those have very specific amino acids that substitute the normal nucleotide sequence of those patients and of those viruses, and we need to use substitution for those type of variants. Then we have the concept polymorphisms. Polymorphisms are changes in the RNA sequence, just as we see in the substitutions, but not all the polymorphisms are associated with resistance. So these are not terms or terms that you should use interchangeably. And finally, the term mutant usually implies selective pressure, which suggests that you are treating a patient with a drug, and because you're using that type of drug, the patient goes into a mutation. So if we understand these concepts, then it will be much easier to go into the next part of my slides. As you remember, the hepatitis C virus circulates in a swarm of viruses called quasi-species. Now, if you analyze the concept of a genotype, we have 6 or 7 major genotypes, and those genotypes have a variation in nucleotide sequence of about 20 to 25%. If you analyze subtypes, and we have about 90 subtypes, the variation of genotype sequences is about 10%. But when we talk about quasi-species, we are talking about genetically related populations. The different nucleotide sequences are probably no more than 3%, and they have different sizes. Now, those relative sizes of the genetically related populations that we found in a swarm of viruses that circulates in someone that is infected depend on the fitness. And the fitness is the replication capability that those variants have in the environment. So therefore, we can see quasi-species that are more dominated, for example, by the red variant, or they may be dominated by the white variant or by the pink variant, while always the dominant variant, in this case in green, is the one that gives the qualification of that group of viruses that are circulating in those quasi-species. So let's now analyze each and every one of them and try to define them. This is the so-called a sensitive variant. The color is totally arbitrary. So this is a sensitive variant. As you can see in this sensitive variant, you have absolutely no amino acids reducing the susceptibility of this variant to DAAs, and we call it a sensitive variant. We can have also sensitive variants that have amino acid sequences, in this case defined by these squares, that are called fitness-associated substitutions. These sensitive variants, we have more replicative fitness. That means they will replicate more readily and will be able to resist more. And as such, those sensitive variants will be very prominent in this type of patients. The third group are resistant variants. As you can see in this picture, I introduce a resistant associated substitution. This variant will be resistant to the effect of DAAs. There are resistant variants that can be unique or single, or they can be multiple. And this is something that we can also detect. And finally, we can have resistant variants that will have also fitness-associated substitutions. In this case, the RAS is in red, and in blue or brown, or I don't know what color is that, is the FAS, or the fitness-associated substitutions. That means that this patient that have this resistant variant will have not only resistance, but also fitness. And as a consequence, that resistance will tend to have more ability to replicate and to perpetuate its presence in the blood. And this is the very beginning of understanding what resistance is. So let me then use this in practice. If you have a quasi-species and you start DAAs, and it happens that the predominant of those that are sensitive are there, most probably the DAAs will eliminate the entirety of the quasi-species, and the consequence of that will be cure, and the patient will be cured. However, if when we introduce the DAA, we start selecting areas that are resistant, or they have not only resistant but fitness amino acid changes, we will probably have patients that will have either breakthrough with predominant of those resistant variants, or even more resistant variants that will lead those patients to develop relapse. And all what I just said can be measured in the lab. This is something that we can measure. So today, we are in the unique ability to analyze all the patients that we follow, determine the resistance that they have, and determine exactly how they are behaving. What's the dynamic move that these patients they have while we treat them. Another concept that are important when we analyze resistance is the concept of genetic barrier. When we talk about genetic barrier, we are talking about the number and type of nucleotide substitutions to generate a resistant associated variant. And now you understand what is, a resistant associated substitution. And the number and type of RASes for a viral variant to acquire full resistance to the drug. That means those patients that will have RASes, but also will have fitness-associated substitutions within the resistance, will be by far more resistant and will tend to perpetuate. The second concept is fitness of the resistant viral population. The more fitness that variant that is resistant is, the more it will perpetuate, and the chances that we'll have problems in the resistance to that particular DAA will be higher. And the final concept is the exposure to the drug. Here is very important to define that we can measure the inhibitory concentrations of the drug in vitro, and we define this by the EC50, and we can determine what drug was applied, for how long, and if the virus was sensitive enough to the effect of that drug. So for example, if we want to use one example of genetic barrier, we can then use, for example, the variant R155K that is a codon of the hepatitis C protease NS34A. And if you have the subtype 1A, that's the genotype 1A, and the wild type has a genotype sequence of AGG and has a change to AAG, we will go from R155 to R155K, and in only one step or change, we will have the mutation. That's why patients that have the subtype 1A can very easily become resistant as opposed to, for example, subtypes 1B. And if you analyze the subtype 1B, you will find that the wild type will be a CGG that will change into the AAG to develop the resistance. But in order to get to that resistant associated variant, you have to have two steps, and that's what we see in subtypes 1B. And that's why you can understand now why certain subtypes are more prone to develop resistances as opposed to others. This is a slide that shows you the antiviral activity, or the EC50, that different drugs have against different variants of the virus. You can see that the lower the EC50, the more sensitive this genotypes in a model of the replicon will have to the effect of the drug. In the first line, you can see how glecaprevir, it's tremendously strong, or the replicon cell lines are tremendously sensitive to the effect of glecaprevir and voxilaprevir, which are precisely drugs that we're using now to treat patients that have resistant variants. If we now analyze just for the NS3 RASes for genotype 1A and genotype 1B, you can see again how the sensitivity to, or the sensitivity of these mutations to the effect of glecaprevir or voxilaprevir is tremendous, while if you compare these same mutations against grasoprevir, paritaprevir, or cimeprevir, they are tremendously resistant. And as you understand these concepts, it's very easy for you to manage the concept of resistance and viral disease. In order to assess resistance, we can use techniques that measure DNA. We can use them in populations of patients that have all these quasi-species. And we divide these techniques in basically two major techniques. One technique called the population sequencing techniques. Some people also call it the direct sequencing, and this is used by the Sanger methodology. It has a sensitivity of about 10 to 25%, or we can use deep sequencing, also known as ultra deep sequencing or pyrosequencing, with a sensitivity of 1%. The problem of these techniques is that these are techniques that are used in virologic laboratories. We use them in our lab, but they are homemade techniques. They are not adequately homogenized, and therefore comparing head-to-head studies using techniques that were using different primers is not very simple. Now there are some commercially available techniques, and perhaps just for your understanding, and by consensus, the 15% cutoff report the presence of resistant variants. Any cutoff under 15% or on any number under 15% is not clinically significant. Just to give you an example, this is a study presented by Purdad in EASL in 2016. It is showing you what is the detection threshold. In this case, they use ultra-deep sequencing, and you can see the number of NS3s, NS5As, or both NS3 and NS5As. And I decided to introduce into this a Sanger methodology for comparison. And you can see that at a 15% consensus, you can see how the numbers go up and give you a more clear idea of what is the sequencing cutoff that we need to determine what drug is useful and what drug is resistant. In this slide, and I don't want to bother you with a lot of molecular biology, but I'm giving you the NS3 protease, the NSA, and the NS5B different amino acid sequences, and I'm giving you in an oval that is orange those that have more than 10% resistant variants, and in green those that have more than 10% resistant variants that are influence the sustained virologic response in each and every one of these subtypes of the virus. So how can we just land all this information into something that we can use? This is the AASLD 2017 giving us some resistant testing in clinical practice. If you are planning to use sofosbuvir, velpatasvir for 12 weeks, then you should recommend in patients with genotype 3, those that are treatment experienced, plus or minus cirrhosis, and those that have treatment naive with cirrhosis. If the Y93H positive, then you should add to the treatment ribavirin. And that's the way you will use this information. If you want to use sofosbuvir, ledipasvir, then you should consider this treatment for someone that is genotype 1A, that is treatment experienced plus minus cirrhosis, and if you find more than 100-fold resistance and the patient has no cirrhosis, you will add ribavirin or use a different regimen. And if the patient has cirrhosis, you will include a treatment of 24 weeks with ribavirin or use a different regimen. I do remember that Rene presented a case yesterday, and Hugo said, I think that in a case like this, I will either check for resistance, which was right, mark for Hugo, and second, he said, I think that I would have treated that patient for 24 weeks and with ribavirin, which again adds a mark for Hugo. And that explains why Hugo is one of the professors of this course. If you are planning to use elbasvir and grasopravir for 12 weeks, which is recommended for patient, and your patient is a genotype 1A, either treatment naive or treatment experienced, you will increase the treatment for 16 weeks and add ribavirin or a different regimen in order to get an appropriate response. And if you are using sofosbuvir, daclatasvir for 12 weeks or 24 weeks, and if you have the RAS Y93H, then you need to do the appropriate intervention as this slide is showing you. So after we have understand what DAAs are and what is resistance, I think it's kind of easy now to try to understand where are we sitting in the world of treatment those patients that have hepatitis C that recur after the first treatments that we use. Now, this is a graphic that shows you from a study that was done through surveillance in population of patients, 6,000 different files from patients of hepatitis C that came from about 155 doctors. They classify what type of patients they saw in their clinics. Now you can see that the majority of the patients were seeing patients that were treatment naive that did not have cirrhosis. And 11% of them were treatment naive with compensated cirrhosis. And 11% of those patients were patients that had recurrence. The organizer of this meeting asked me to talk precisely about that 11% of the patients that experience treatment recurrence because they are treatment experienced. So what is hepatitis C recurrence? How frequent is hepatitis C recurrence? We are talking that the success of this treatment today varies per genotype, but easily goes from 85 to 99%. That means that between 1 and 15% of our patients will still have recurrence. And that recurrence is still a matter of questioning and investigation these days. So if we analyze this overlapping circles, you can see that we have the host in one hand, the virus in the other hand, and the disease in the third hand. And the concepts or elements that can generate hepatitis C recurrence is that some of these hosts may not be very good metabolizers of the DAA, that maybe they have a very specific genetic status that will make them more resistant to the effect of the treatment, or more simply that probably they will not have, they are not adhering adequately to the treatment that we want to use. If we are now focusing on the virus, we understand that there are resistance to different viruses, and if we want to focus on the disease, we understand that those patients that have fibrosis or cirrhosis will have a tendency to have a disease that will be more resistant to the effect of treatment. This is a, yeah, can I have the mobile thing, because otherwise I cannot see your faces. When you analyze this graphic, this is from the Italian Resistance Network, you will see that the prevalence of failure in their series is 81% when the patient had cirrhosis, 40% when the patient had genotype 1 or genotype 3. Gracias, Jimena. When they viral load was more than 8 million, when they were treatment experienced, or when they have multi-RASes in the sequence of those viruses. Now, if you analyze the outcomes by previous treatment regimens, you can see this is a study that used the combination of grazoprevir and elbasvir. You can see patients that were treated with this combination were treated with boceprevir, telaprevir, and cimeprevir. And you can see the different RASes that those patients had. Back in those days, we did not call them RASes, but RAVs, or resistant associated variants, but this is representing the RASes. As we treated those patients with a combination of grazoprevir, elbasvir, the sustained virologic response in this patient was 95.5%, and for those patients that had prior non-virologic failure was 100%. Obviously suggesting that we have treatments today that are very effective for those patients that were resistant to prior treatments with NS3-4As of the first generation. This is a study that used sofosbuvir-velpatasvir plus ribavirin for 24 weeks for those patients that were resistant to the effect of the same medication, but only given for 12 weeks and without the effect of ribavirin. And you can see here how those patients that did not have RAVs in blue, 82%, had a sustained virologic response of 96%, and this 18% of patients that have RASes, they have a response of 100% just by increasing the treatment to 24 weeks. That is, by increasing the EC50 by giving more medication in association with ribavirin. If you analyze patients with genotype 2, again, the presence or absence of RASes had no effect on these patients. That is, genotype 2 patients have a very nice fitness for sensitivity. But once you go to patients with genotype 3, even treatments for 24 weeks in association with ribavirin only gave a sustained virologic response of 77%. That means that if you have a patient that is a genotype 3 that is resistant to sofosbuvir, velpatasvir, you will need to consider treating this patient with something else that just increasing to this medication for 24 weeks and adding ribavirin. And that's where we came to the last generation of medications against hepatitis C, and that is the use of the combination of pibrentasvir, glecaprevir, a NS5A with an NS3 4A. And in this study presented in EASL in 2016, you can see the different regimens that were analyzed, the resistant variants that those patients have. And in this slide, you can see that the sustained virologic response achieved with this treatment was almost uniformly around 90 to 100%, independently of what type of RASes those patients have, suggesting that this is a great treatment to be used in those patients that are resistant to treatment. This is another option. This is a triplet option, combines sofosbuvir, velpatasvir, and voxilaprevir, 3 drugs. You can see that they use this in patients that were NS5A treated, 51% of those patients with ledipasvir, 27 of them with aclatasvir, 11% to ombitazvir and other medications. And as they were treated, the response rate, the overall response rate was 96%, in those without cirrhosis up to 99%. And when you analyze this treatment, depending all the different genotypes that were used, again, the response rate was very close to 100%. If you now analyze the different patients according to the RASes, you can see that those patients that did not have any RAS, 17% had a sustained virologic response of 98%, but perhaps more importantly, those patients that have NS5A resistance, NS3 plus NS5A or NS3 only, when they were treated with this triple combination, had a sustained virologic response of 96%. Again, telling you that we have expanded the armamentarium of treatment of hepatitis C to drugs that are tremendously effective in this treatment. So if today you want to take home some retreatment recommendations, and these are recommendations by the ASLD, if you have a failure to NS3 PIs, protease inhibitors, with pegylated interferon and ribavirin, and your patient is genotype 1, you can use glecaprevir, pibrentasvir, sofos, velpatasvir, sofos, ledipasvir for 12 weeks if the patient has no cirrhosis or compensated cirrhosis, and your chances to get a sustained virologic response close to 100% is pretty good. If you have a patient that has failure to non-SS5A and a regimen that contain an NS5B like sofosbuvir and genotype 1, then again, you can use glecaprevir, pibrentasvir, or the triplet of sofos, velpatasvir, voxilaprevir for 12 weeks, independently of the presence or absence of cirrhosis, and you would expect to have a very good treatment response. If you have a genotype 2 and you had a failure to sofosbuvir, ribavirin, which we see in Mexico, we use glecaprevir, pibrentasvir, or sofosbuvir, velpatasvir for 12 weeks, and you will have a very good response. If you have a failure of NS5A, G1, then you use the triplet of glecaprevir, pibrentasvir, and you do adjustments with time. If you have retreatment recommendations for patients that are genotype 3, and this is a tough genotype, then you use the triplet of sofosbuvir, velpatasvir, voxilaprevir for 12 weeks. And if you have a patient with compensated cirrhosis, then you add ribavirin. And then, dun-dun. Thank you very much.